Okay, so just a continuation of our cytoskeleton lecture. So this is part 2. We've talked about actin and myosin, and now we're talking about microtubules. So again, remember that the cytoskeleton itself has 3 main components: actin filaments, microtubules, and also intermediate filaments. And then the myosin interacts with the actin and is really there to provide sort of a means for movement along the actin filaments. Okay, so we talked about myosin. So now we're talking about microtubules. So these guys have a slightly different structure than the actin filaments. They're hollow, okay, and they're pretty rigid. They too are also dynamic, so they undergo continual assembly and disassembly, just like when we talked about actin, we talked about polymerization and depolymerization. This is the same idea, okay? And same thing, they're going to function in cell movements and also determining cell shape. So there's a lot of overlap in structure in terms of microtubules and actin. Remember, we talked about actin cytoskeleton really as being localized in the periphery of the cell, right? Or this cortical cytoskeleton near the plasma membrane. That's not necessarily the case with microtubules, okay? All right, so if we talk about assembly, again, this is very similar to what we talked about with actin, but also, slightly different. So you'll have microtubules made from tubulin, okay? And they'll form dimers that will then polymerize, okay? And then you'll also have these, and so these protofilaments that are formed will basically be arranged in parallel, okay? And just like actin filaments, these guys have polarity, okay? There are plus and minus ends. When we talked about actin, we had barbed and pointed ends. It's the same idea, okay? All right, so again, if we talk about what they're made up of, globular protein tubulin. Again, we talked about actin being made up of, or actin filaments being made up of globular actin, right? Okay, so again, the tubulin form dimers, okay, and then they'll form these sort of filaments from there. Okay, so if we look here, you can see again these are the globular tubulins that join together to form the dimer, and this is referring to the beta and the alpha tubulin. Again, I'm going to ask you about the structure, and even just knowing that it's beta and alpha tubulin, it's not really that important at this point. We really just want to know that they form dimers, and then they arrange to form these sort of hollow tubes. Okay, again, if you want to look at it in a different way, you can see this model, and again, you can see that just like when we talked about actin associated with either ATP or ADP, here you're going to have tubulin, binding to or associated with GDP and GTP. Okay, so same idea. The binding of either GDP or GTP is going to alter the activity or the binding activity of the tubulin. Okay. Okay, so now let's talk about the assembly and the disassembly. So again, GTP and GDP are going to affect binding, okay? So let's look at a picture. So again, this is pretty much describing what's in this next picture here, okay? And again, similar to what we talked about with actin. So if we're talking about microtubule polymerization, you look at the plus end versus the minus end, right? You can see that growth is occurring at the plus end. These, tubulin proteins are bound to GTP, okay? And that's going to basically make them more apt to bind to each other, okay? And therefore, that's why these are sort of attracted here to this plus end. However, if you look down at this end, the minus end, these guys are GDP bound, okay? They have less affinity for each other. They essentially fall off, okay? All right. Again, so we called when we talked about this with actin, we called it treadmilling. Okay, again, it's kind of the same idea, but it's not treadmilling. It's slightly different. But again, you can see a lot of similarities here. Okay, so there's a term that we use called dynamic instability. Okay, so it says microtubules that are stabilized at the minus end. In microtubules that are stabilized at the minus end, if you have rapid GTP hydrolysis, basically you can have something called this dynamic instability, and those tubules, the tubulin molecules breaking away from each other, okay? And so the GTP and GDP, again, is a way of the cell, one way the cell can regulate whether they want these microtubules to, grow or maybe disassemble, okay? And so it's a way of regulating the assembly and the disassembly of the microtubules, okay? All right, so again, as long as new GTP-bound tubulin are added more rapidly than GTP is hydrolyzed, then it's going to grow, okay? If GTP is hydrolyzed to form GDP more rapidly than the new subunits adding, then you're going to get disassembly and shrinkage. Okay, and again, this is on purpose. We have regulation of the GTP and the GDP on purpose, depending on what's going on in the cell and what needs to happen. Okay, so again, this is showing you, I mean, it's kind of similar to what we saw before, but if you're talking about growth, right, you're going to have more of these guys being added than you have in terms of GTP being hydrolyzed into GDP. Okay, and you can see that you have growth on this end, and that this is really not, you know, sort of disassembling. Again, if you have a lot of hydrolysis of GTP to GDP, you're going to start having these guys fall off. Okay, so more GTP-bound tubulin is going to lead to growth. If you have increased hydrolysis of the GTP and more GDP-bound tubulin, it's going to cause disassembly. Okay, so we can talk about assembly and disassembly, or growth or shrinkage, okay, however you want to term it. Okay, now, just like we talked about some drugs that affect the actin filaments, there are drugs that particularly affect microtubules, okay? So here are some drugs, and these actually are used, again, in the lab, like I said, but also used in cancer treatments, okay? So drugs like colchicine and colcimid, okay, will affect microtubule assembly. And so, again, and since you guys aren't here, I can't ask you this question, but think about why affecting microtubule assembly would be important in treating cancer. Okay, why would these drugs be used to treat cancer? Other drugs, vincristine and vinblastine, are used again as cancer chemotherapy too, because they inhibit microtubule polymerization. Okay, again, the microtubules, right, are basically the spindle in mitosis. Okay, cancer cells are rapidly dividing cells. If you inhibit microtubule polymerization, you basically can't make that spindle. Spindle can't grab onto the chromosomes. Mitosis is disrupted. The cell does not divide. Okay, and so these are chemotherapy agents, okay, that affect microtubule polymerization. Another one, Taxol, will stabilize microtubules and also block cell division, because again, you have to have this dynamic sort of shrinkage and increasing or growth of the microtubules in order for mitosis to occur, okay? So if you don't allow that dynamic instability, the microtubules can't really function properly. Okay, so given that, right, we're just kind of talking about mitosis, and that if you disrupt microtubules, it's going to interfere with mitosis, right, and particularly the formation of the spindle and also the function of the spindle, okay? Most microtubules will extend from the centrosome, okay? We've learned about the centrosome before. And so if we talk, especially during mitosis, right? So if you talk about during mitosis, these microtubules will extend out from the duplicated centrosomes to form the spindle, okay? So you kind of have the centrosome there. If a cell is going to divide, it will duplicate that centrosome. Those two centrosomes move to opposite ends of the cell, right? Opposite poles, and then you're eventually going to have microtubules extending out, okay? And so just a reminder, we should all know this, okay? Again, here's your centrosome, and you can see microtubules extending out. If you're talking about a cell that's going to undergo mitosis, centrosome duplicates, and then you have the spindle forming, you know, and the centrosomes on opposite ends of the cells. These purple guys are microtubules, okay? So anything that's going to disrupt these microtubules is going to affect mitosis and the formation of the spindle. Okay. So just a little more about the centrosome, okay? So again, the centrosome exists in an interphase cell, in a cell that's not going through cell division, okay? It's duplicated during mitosis or right before mitosis and then forms the spindle, but it's there, and there are microtubules obviously in cells that are not dividing, okay? So it says that the centrosomes are initiation sites for microtubule assembly, okay? So essentially, where the microtubules are going to start forming are at the centrosome. Okay, so like if the cell had no, and I'm going to show you a little picture that sort of illustrates this, but if the cell had no centrosomes in it, right, and now it's going to start forming microtubules, it's going to start, or sorry, if it had no microtubules in it, but now it needs to form microtubules, that assembly is going to start at the centrosome. Okay, So it says if you treat cells with colsamid, okay, which one of the drugs we just mentioned before, the microtubules will disassemble. Okay, so treating a cell with colsamid will basically cause all the microtubules to disassemble. Okay, if you then remove that drug and allow the cell to recover for about 30 minutes, you'll see that the microtubules grow from the centrosome. Again, this illustrates the point that that centrosome is basically where microtubule assembly is initiated. Okay, so here's your normal interface cell, and what this is stained. So what you see that's bright is it's stained for microtubules essentially. Okay, so you're looking at just microtubules here. If you treat it with colsamid, you can see that you lose all that microtubule structure, right? And then what you see is now they're reforming from the centrosome. Okay, so that's what that's showing you here. Kind of just a side note, if we go back here, in fungi, okay, there's no centrosome, right? Spindle pole bodies have a similar function to the centrosome. Again, plant cells don't have centrosomes, but microtubules still form. They just don't form from the centrosome, okay? All right, so we saw that already. Now, just like actin filaments had actin binding proteins, microtubules have microtubule-associated proteins. You can call them MAPs or MAPs, and these guys will be involved in regulating that dynamic behavior of microtubules, right? Whether they're going to grow or whether they're going to shrink. Okay, so it says minus ends are stabilized by proteins that prevent depolymerization, right? So at that minus end, there are certain microtubule-associated proteins that will bind there to stabilize them, okay, so that they don't depolymerize. It says growth or shrinkage of plus ends is regulated by other microtubule-associated proteins. Bottom line is there are microtubule-associated proteins are proteins that bind to the microtubules that either maybe stabilize them or initiate growth or shrinkage, what have you. Okay, and so again, just like we had this table with the actin binding proteins, I don't expect you to memorize these. We will talk about a select few. Those are the ones you should know, but you don't need to know this whole table, okay? But you can see, again, there are certain, and again, this is not all of them. There are many more. But you can see that sort of each microtubule-associated protein has a particular function, okay? So like kinesins involved in plus end depolymerization, right? So meaning they're gonna basically cause that end to depolymerize, okay? So they can kind of come in and sort of change that sort of activity of the microtubule or the structure of it, okay? If you talk about tau, which we're going to talk about a little bit, these guys are going to stabilize the microtubules, okay? Okay. All right, so we talked about this. So they allow cells to stabilize microtubules. I just mentioned that. One in particular, I mentioned tau, right? And help determine cell shape and cell polarity. Okay, so these are involved in cell shape as well, okay? I already mentioned that you have many different types of microtubule-associated proteins, and many of them are cell type specific, meaning you're only going to see this certain microtubule-associated protein in a neuron, and you would see, a different one in another cell type, like a, I don't know, whatever, a skin cell or something like that, okay? So one example is tau, like I mentioned before, and this is a protein that's characteristic in brains of Alzheimer's patients. We mentioned this when we talked about misfolded proteins and prions and things like that, okay? But you get these aggregates of tau. This is also super important when you talk about CTE, chronic traumatic encephalopathy. So this is, caused by repetitive blows to the head. Okay, you'll see this sort of accumulation of tau, and again, you get these aggregates. Okay. All right, so we're mentioning neurons, right? So microtubules are super important in neurons, right? I mean, again, think about the shape of a neuron, not like a regular cell. A lot of things need to be moved around. I mentioned this kind of before, right? That you need to sometimes move things from the cell body all the way down to the axon terminal, okay? There are also things that just need to move around cell body in general, right? But they're particularly, these are one particular example where you can see how important microtubules are, okay? So nerve cells actually sort of have two kind of different types of microtubule-associated proteins, okay? So in axons, okay, the microtubules have plus ends towards the tips. We're going to see a picture. And these microtubules in the axons are associated with tau. Okay, we just mentioned tau. In the dendrites, however, Microtubules are oriented in both directions, kind of meaning that you can have movement in both directions, meaning movement away from the nucleus towards the cell periphery and towards it. When you talk about the axons, really most of the movement is going towards the axon terminal, okay? And in the dendrites, the microtubule-associated protein is MAP2, okay? So tau is located in the axon, MAP2 is localized to the dendrites. Okay, so if you look at a picture, here's our neuron here, right? Cell body, these are dendrites, this is the axon, and these are axon terminals. Okay, if you zoom in here, now you can, right, here's the nucleus, you zoom into the nucleus, you can see here in the dendrites, again, these are processes, right? These are protrusions or extensions of the cell that have particular function, and things need to be able to move, you know, in through all of these extensions, okay? And so you can see here, these are microtubules in purple, and you can see this MAP2, which associates with these microtubules. These are other proteins that help stabilize them as well. Here you have tau, okay, that's bound in the axon, okay? So you can see this different localization of MAP2 versus tau, okay? Let me just mention a little side note too here. So I mentioned CTE before. So if you talk about traumatic brain injury, it can lead to this CTE. And one of the hallmarks of CTE is, again, these aggregates of the tau protein. And so what happens is you basically can have this tau protein get hyperphosphorylated, right? So remember we talked about phosphorylation as being a regulator of protein activity, okay? And so if this tau protein gets more phosphates added to it than it should, it causes misfunction, and then it causes the tau proteins to dissociate from the microtubules. Once that happens, these become destabilized, okay? And then it becomes destabilized, and then essentially the axon becomes destabilized. The neuron can't function if it loses its connections here, and basically the axon is not functioning properly, and the neuron ends up going on to die, okay? So that's how tau is sort of associated here. So again, if tau is not here to stabilize these microtubules, it'll just totally lead to neuron dysfunction and neuronal death. Okay, so and just like it's sort of illustrated here, right, things are going to be able to move along these microtubules, okay? And so again, we talked about actin filaments and myosins that move along the actin, right? With microtubules, the motors are different proteins, but it's the same idea, guys. This is all similar in terms of ideas, right? So you have microtubule motor proteins, okay? And these are, some examples of them are kinesins and dyneins, okay? So kinesins will move along microtubules towards the plus end, and dyneins will move towards the minus end, okay? And again, a major role of microtubules in the cell are to transport things around, okay? So yes, things can move along actin filaments as well, but a lot of the transport or microtubules are predominantly involved in transport of macromolecules, of vesicles, of organelles, of a whole bunch of things, okay? We did mention that things move along the actin filaments as well, like vesicles, right? But I think if you look here, the microtubules have a sort of more extensive role in moving things around the cell, if that makes sense, okay? And again, the way this is done is that these kinesins and dyneins will transport cargo, okay, along the microtubule. So again, this looks very similar to what we saw with myosin and actin, right? And except here, it's microtubules, and this is dynein, right? And this is kinesin. It's very, very similar. Again, these are just proteins that bind to the microtubules on one end and then bind to some cargo on the other end and move things around, okay? So again, I would think of, you know, if you want to try to separate functions of actin cytoskeleton versus microtubules or actin versus microtubules are definitely more involved in transport through the cell, okay? Actin is as well, but really microtubules even more, okay? All right. Now, again, like I said, there's a lot of overlap between microtubules and actin, okay? So there are some projections. We kind of called them protrusions when we talked about actin, but there are also cell projections that are microtubule based, okay? And these are ones that, you know, like flagella can move, right? And so kind of if you think about these as being, I guess, more a little more dynamic than something like a microvilli. However, we also saw examples of, you know, actin protrusions like in phagocytosis where it's dynamic as well. So So anyway, so maybe that's not the best sort of comparison, but bottom line is cilia and flagella are microtubule based, okay? These, again, these are protrusions from the cell surface, and but again, if you look at cilia and flagella, these are kind of moving in a sort of, I don't know, regulated or constant. It's kind of constant movement, okay? Whereas if you talk about like phagocytosis and things like that, it's not this constant kind of movement. So slightly different there. Maybe that helps. I don't know. Cilia, okay, will be sort of move in a coordinated back and forth motion, okay, which can either move a cell through fluid, right, or it can move fluid over the surface of the cell. So if you think about cells that line the trachea, as air is coming in there and there's water in there and particles, those cilia can actually trap things and then get it back up. So they're kind of catching what's there. There are ciliated, single-celled organisms that move around via cilia. There are ones that use flagella as well. An example of cells with flagella in our bodies are sperm cells, okay? Super important that sperm cells have good motility, okay? And you can see this here, and that helps them move, okay? And this is an example of showing you actually a ciliated cell. So I gave you an example of a ciliated cell, cells that line the trachea. All right, so that's for microtubules. So for microtubules, we talked about their assembly and disassembly and the idea of dynamic instability and that they have a plus end and a minus end, right? There are drugs that can block microtubule assembly, and a lot of those drugs are used for cancer treatments and even in chemotherapy, okay? Microtubules are super important in formation of the spindle, right? And that they form out of the centrosome. Okay, and then we talked about movement along the microtubules. Okay, we talked about some microtubule-associated proteins, okay, particularly in neurons, tau and MAP2. And then, like I said, we talked about movement along the microtubules, and two proteins that we mentioned that act as sort of microtubule motors to move things around were kinesins and dyneins. Okay, and now we talked about cilia and flagella, okay, these sort of modifications of the cells that are microtubule based, okay? The last component of the cytoskeleton are intermediate filaments, okay? And again, they're called intermediate filaments based on size, okay? So they are intermediate between actin and microtubules, okay? So that's where their name comes from. They are not directly involved in cell movements, but they do basically provide mechanical strength, okay? So really microtubules and actin are the big players in cell movement, in movement of things around the cell, in cell shape, okay? Intermediate filaments, not really, okay? They're really there to provide sort of strength and scaffolding, okay? And you don't see intermediate filaments in yeast, plants, and also even some insects don't have intermediate filaments in their cytoskeleton. So the cytoskeleton would just be actin and microtubules, okay? These guys are composed of many different types of proteins. So when we talked about actin and microtubules, right? Actin was composed of actin. Actin filaments composed of actin. Our actin cytoskeleton composed of actin. Microtubules composed of tubulin, okay? Here, the intermediate filaments are composed of many different types of proteins, okay? So here are some examples of some proteins that are considered intermediate filaments. So type 1 and type 2 are keratins, okay? So in epithelial cells, you'll have keratins or keratin proteins that make up these intermediate filaments. Also, vimentin, okay, will be another protein that's part of intermediate filaments, okay? And again, you'll see that you'll have particular intermediate filaments depending on what type of cell you're talking about, okay? So we mentioned keratins, epithelial cells, okay? If you look down here at neurons, there are particular types of intermediate filament proteins like internexin that are in neurons, nestin in stem cells, okay? So you can actually label cells with nestin. You can use an antibody to determine if they're stem cells. And this is actually used a lot in neuroscience research to determine neuronal precursors or neuronal stem cells. Okay, so anyway, you can see that there are many different types of intermediate filament proteins. Here's another one, GFAP, is in glial cells. So these are supporting cells of the nervous system. Same thing, you can label if you have an antibody and then you basically have it tagged with some sort of fluorescent tag, you can visualize glial cells that localize that GFAP, okay? So again, many different proteins that are considered part of the intermediate filaments. This is really just like super short. I think we have like one more slide. I'm not getting into many of the details here, but I guess just to kind of bring in some of these experiments and how we, figure these things out. So it says in terms of the role of intermediate filaments, one of the ways we've learned about the role of intermediate filaments was using transgenic mice. So these are mice that have a mutation in that keratin gene, right? Or in one of the keratin genes. I'm not exactly sure exactly what these were, okay? Right? So you basically mutate that gene and you grow these mice up to have a mutation in, a keratin gene. Okay, and then we look to see what sort of deformities or dysfunction occurs in the transgenic mice. Okay, so if you want to find out about the function of something, you can knock it out, right, or just disrupt it. So it says the mutation disrupted formation of normal keratin cytoskeleton, obviously, and it actually particularly resulted in skin abnormalities, okay? Because we said that keratins were particularly expressed in epithelial cells, okay? And so this is basically just showing you, right? If you're you can basically have your mutant keratin, you introduce it into, cells that are going to develop. Basically, this is it, right? Fertilized egg. So you're basically injecting it into here, and as these cells basically divide and start developing into an embryo, they're going to have this mutant keratin, okay? And so now these pups, some of them will grow up to have, a mutant copy of this mutant keratin. Some will be normal, okay? So this is basically showing you here. So this guy that expressed this mutant keratin, you can see that he had skin blistering and severe skin abnormalities just due to a dysfunctional keratin protein, okay, which again is predominantly part of the intermediate filament component of the cytoskeleton. And that is the last slide. Okay, so I will now upload this as part 2. All right, thank you guys. And remember, be on the lookout for the quiz because it's going to be coming. Okay.